So now that we've seen some evidence from the fossil record for how speciation occurs, and specifically in relation to its rate, we can continue that discussion of the rate of speciation in this next flowchart, and we'll entitle it Rate of Speciation 2. In this flowchart on speciation, we're going to continue that discussion on how fast it occurs, but more specifically, we're going to be looking at a direct example, a lab example that can be seen and has been observed by scientists to show how fast speciation can happen. One thing we have to understand about the rate of speciation is the following. Once speciation begins, okay, so we'll write that down. Once speciation begins, it can be completed rapidly. It's just sort of a side effect of once it begins, there's this option for speciation to be completed rather rapidly. And we're going to be seeing that with the example that we put forth. Furthermore, this is exemplified by the following lab study. And that study was done on an organism known as Helianthus. That's the genus, and the species is Anomalous. This is a type of wild sunflower, so we're going to call this the wild sunflower. And what we notice about this wild sunflower that's quite interesting is the fact that it actually originated from a hybridization, let's say. So it's a hybrid. So it's the origination from a hybridization of two different species, okay, of two different, let's say, sunflower species. And this means that we have certainly in this species a reproductive isolation event. It is the result and eventually leads to reproductive isolation, and thus it is its own species. So Helianthus anomalus is its own species because it is a hybrid of two different sunflower species that reproductively isolates itself from the parent species. So what is the experiment that shows us the rate of speciation with this model organism? So let's look at that experiment for the rest of this flowchart underneath this arrow. So the experiment. In this experiment, we're going to start off with this background information. What we have is two species of the type that are going to create Helianthus anomalus are going to be crossed. So two species were crossed, the ones necessary to create this hybrid, and each of these species had 34 chromosomes. So we have the same amount of chromosomes, each with 34 chromos, and this results in the following. When we have this cross of two different sunflower species, we get Helianthus, we get an F1 hybrid, known as Helianthus anomalus, and now we want to ask the question we always are wondering about this F1 hybrid. What is going to happen to it? What we notice is that F1 hybrid has 1/2 of its chromosomes, so we're going to write this down again. One F1 hybrid, it has 1/2 of chromosomes, so 1/2 chromo from that one species, okay, from, let's say, 1/2 chromo, one species, and we'll say that the other half of those chromosomes, so there's 2 halves, from the other species. So look, two species were crossed, each with 34 chromosomes. The F1 hybrid, the combination of both of those, has half the chromosomes from one species, half the chromosomes from the other species, thus it's a hybrid. Okay, so what we're going to do with this knowledge and this information is do a simple test. And the test will involve the following. We're going to test the fact and ask ourselves, are the F1 hybrids, so are the F1s, and its subsequent generations, G-E-N-S for generations, fertile? Are they fertile? Can they produce offspring? Because what we've noticed for the most part is that hybrids aren't able to produce offspring. And because they're not able to produce offspring, we don't have reproductive isolation completely, and thus we have a post-zygotic isolation mechanism, let's say. So we're going to ask ourselves, does this happen in this species? And what we notice, and we do this through the comparison of chromosomes, we actually have this ability to compare, eventually, the chromosomes of the species in question. So let's say the experimental hybrids. So we can take a look at the experimental hybrids and its chromosomes. And why are we looking at the chromosomes? Well, the chromosomes are what will be passed down. And if the chromosomes are okay, then the species that is going to develop will probably be okay. So compare chromosomes of experimental hybrids plus the chromosomes that are present in the natural Helianthus, let's say species that we're looking at, and we're going to compare and contrast these. And this is going to be done by creating what we call species-specific genetic markers. So I'm going to put a genetic marker on the experimental hybrid chromosome. Let's say those chromosomes are going to light up blue if I put it under phosphorescent light. And if we look at the naturally occurring Helianthus, they light up red or something, those chromosomes. And we're just going to compare and contrast and see, is there a difference? And if there is a difference, what is causing the difference and why? Well, the results from this experiment are the following. So let's see the results. We have a test. Are F1s and subsequent generations fertile? So are these guys, F1 hybrids that I made here, fertile, and are the F1 hybrids children fertile as well? The results state the following. Only 5% of the F1 are fertile. So only 5% F1 equal to fertile. So that's quite a low number. But where it gets really, really weird is that if you continue having these F1 offspring to continue their generations, mainly we say after about 4 generations, we notice something very, very weird. So after 4 generations, fertility actually rose to about 90%. That is a huge, huge spike. So fertility rose to 90%, meaning that 90% of the, let's say, descendants of this F1, who originally only 5% of them were fertile, 90% of them after the 4th generation were fertile. That is a huge 85% increase over a short period of 4 generations, which can be seen and is established in the lab. And then once we get to the 5th generation, we notice a big sort of conclusion. In the 5th generation, we notice that the difference between the hybrid and the F1s, there is still a bit of a difference. So F1 versus hybrid are slightly different. So they're slightly different. What we mean by this is that those of the 5th generation versus those of the 1st generation are a bit different, but they are actually going to be rather similar. We'll say rather similar to the natural, Helianthus. So we've gone and deviated from our parent population so much so originally in this F1 hybrid, but after 4 generations our fertility rose all the way to 90%, and by the 5th generation you could easily say that we have a completely new species altogether, it seems, because we have reproductive isolation from that F1. The F1 only has 5% fertility and this has 90%. That is a huge, huge difference, and thus we have essentially created a new species. And so we can finally end this video and this flowchart on some basic conclusions from this study. What we can state is that over time, hybrid chromosomes we notice from this experiment, so we'll say over time, the hybrid chromosomes, the ones that were in question, remember how we had to genetically mark them over here? Hybrid chromosomes, they sort of developed, they got better over time. Over time, hybrid chromosomes became more similar to natural. i.e., these chromosomes started becoming more and more fertile, becoming more and more viable, becoming more and more successful and able to promote fertility. And because we have this promotion of fertility, we're going to have a new species. And we're going to ask ourselves, why did this happen? Why did we have these chromosomes developing better and better with each generation? Well, we can state that selection itself is going to eliminate those that were bad. Selection literally eliminated what we call, and we've referred to this before, as incompatible genes. those genes that cannot produce viable and fertile offspring, they're going to be eliminated by selection, and thus the chromosomes are going to get better and better and better after every single generation, up until the fifth generation, where we almost have an entirely new species completely. Final conclusion to understand speciation through this experiment, you know for a fact, is a rapid and observable process. It's rapid and it's also an observable process because this was all very clearly seen within a laboratory controlled environment, so thus speciation is certainly possible, and thus we can definitely see on a large scale how macroevolution events can clearly happen over a much longer period of time, which would mean that we have much more speciation, which would make a lot of sense because we have so much variation within the natural world.